All right. Well, hello, everyone, and welcome to the University Scholar Series. It's so exciting to be here today and have the opportunity to hear from one of our outstanding faculty members, Alberto Rascón Jr., who's going to be talking about a quest to control the female Aedes aegypti mosquito population, which I'm really looking forward to this talk. Before I get to a little bit of background, I just want to do a couple of housekeeping issues. So if we can advance to the next slide, just so you know, if you want, there's live captioning available, and also we're recording this webinar so that others who might not been able to be here today are able to take advantage of this opportunity. So please just understand that the recording is happening, but that live captioning is available if you need it. Secondarily, I'll be moderating the questions. So if you have questions, there's a Q&A button at the bottom of the screen. You just click that. You can ask questions anonymously if you like, or you can put your name on them, and I will go through those. You can also upvote questions. So if you see a question you are already interested in asking, you can just like it and it'll pop to the top. So just know to use the Q&A function, and you can start asking questions before he's done, and then we can even start to go through those when he's finished with his talk. So with that, let me just do a little bit of background on our colleague and the land acknowledgement before we get going. It's important for us to acknowledge the land on which we're meeting today as the traditional home of the Petron Ohlone speaking people and the present-day Mamekwe Ohlone tribe. The Petron Ohlone were missionized into both missions Dolores and Santa Clara. The present-day Mamekwe Ohlone tribe is comprised of all known surviving Native American lineages aboriginal to the San Francisco Bay region, who trace their ancestry through the missions San Jose, Santa Clara, and Dolores, and the historic federally recognized Verona Band of Alameda County. In the Mwekwe Ohlone language, Cocheño, Mwekwe means the people. Without them, we would not have access to this gathering. We take this opportunity to thank the original caretakers of this land. With that, then, let me now turn to just a little bit of background on our speaker today. A Californian who was born in LA and grew up in the Central Valley. He completed his degree, his undergraduate degree at CSU Bakersfield under the tutelage, as I understand it, of Carl Chemnitz, now provost, but formerly of our own campus. Alberto went on to get a PhD at the University of Arizona, working with a colleague of mine that I know well, Roger Meesfield, who just speaks so highly of Alberto and his contributions. Alberto's dissertation led him to an opportunity for a postdoctoral fellow at the Center for Drug Discovery and Innovation in Parasitic Diseases at the University of California, San Francisco, before he joined us here. He is now a tenured associate professor with a background in biochemistry in our department of chem. One of his most recent papers, published in PLOS Biology, takes up a really important question about mosquito-borne diseases, which remain a massive killer of many humans today, several million human deaths. And he is looking at, with his research partners, how to manage those populations. And this is a really important question, as we've seen climate change the habitat of mosquitoes today. So with that, I just want to say I'm very excited to have Alberto speaking, and I would like to turn it over to him now to talk about a quest to control the female Aedes aegypti mosquito population. Alberto, it's all you.
Thank you very much. That's a great introduction, and I'm very happy to be here today. I'm happy to share the work that we've been doing here at San Jose State, and as the title indicates, take you through this journey of our quest to control the female Aedes aegypti mosquito population. So what I'm going to do is I'm going to start off with giving you a little bit of background information of why we even want to focus on the mosquito. I mean, Vin kind of touched a little bit on that information. And also, though, the blood feeding behavior and how the blood feeding behavior has led to the transmission of specific viruses. Then we're going to go into some of the work that my students have done here at San Jose State, and then kind of summarize everything together and hopefully convince you that it is worth going after this mosquito. So to start off, let's start off with the Aedes aegypti mosquito life cycle. So the female Aedes aegypti mosquito must uptake or imbibe a blood meal, and typically she will imbibe or uptake a blood meal more than her body weight. So the thing is that the mosquito then, once she uptakes the blood meal from a human host, this is going to initiate the release and activation of proteases or digestive enzymes in the midgut. So this will then lead to the metabolism, pretty much the breakdown of blood meal proteins in the mosquito, leading to the nutrients and the energy needed for the egg laying process. So a word that I really want you to keep in mind at the moment is called the gonotrophic cycle. So when we talk about the gonotrophic cycle, what I'm referring to is the uptake of a blood meal, the metabolism of those blood meal proteins, and the egg laying process. So pretty much this little corner here is the gonotrophic cycle. So once the mosquito lays her eggs, and it's typically done in an aquatic environment, these eggs will hatch into two different aquatic phases. One is the larvae stage, followed by a pupae stage, and almost 7 to 14 days later, the mosquito will emerge or eclose as an adult, and she's ready to mate and then uptake another blood meal. But the problem is that this blood feeding behavior has facilitated the transmission of bloodborne pathogens from the mosquito to human hosts. So many of you will probably already know that the mosquito is a carrier of the Zika virus, dengue virus, chikungunya virus, and the yellow fever virus. And when people become infected with one of these viruses, typically many have little minor fevers. It's almost like a flu, minor fevers, headaches, chills, bone, muscle, and joint pain. But at its worst cases, for example, with dengue and yellow fever, there are stages known as hemorrhagic stages where blood platelets are actually affected by the virus and people will start bleeding easily. But and then also with the bleeding and the bruising, they could also lead to internal hemorrhaging and potentially death. And with chikungunya here on the bottom, people actually typically have very severe joint pain, and the pain is so bad that a lot of people wish they were actually dead instead of surviving with the pain. And then the Zika has been linked to microcephaly in babies. And microcephaly is a case in which the babies are born with underdeveloped brains and also underdeveloped craniums. So there's a lot of motor skills and things like that that are affected. And furthermore, what makes the mosquito very, very dangerous is that she is an efficient biological vector. What this means is that she actually has to acquire more than one blood meal until she's fully engorged. So as you can see here in this picture, that nice little red blood meal. So the mosquito, she has to first become infected from imbibing or uptaking of the virus from an individual who's already been infected. And once she becomes infected, the mosquito can then transmit the viruses to an uninfected human host. And I really wish we could have kind of done this in person, because what I like to give the example is that if we were all in one room and there was an infected mosquito in the room, and if she bit me and if I didn't kill her, she would go on to the next person, to the next person, to the next person. And if no one kills her, she's not going to stop until she actually gets a fully engorged belly. So she could potentially infect about 5 to 10 to maybe 20 people until she actually has this huge engorged belly. And so that's why she's a very efficient biological vector. And I don't want to scare you with the information that I'm presenting or kind of being alarmist, but the thing is that with these viruses, they used to be known as third world tropical diseases, but it's actually becoming more frequent, and the presence of the mosquito is actually in the United States. And unfortunately, there are no vaccines available against the Zika virus, the chikungunya virus, There is a limited vaccine against the dengue fever virus, but it has not been FDA approved. And in fact, people who actually have tried this virus in the Philippines, for example, they actually have shown that people who are inoculated with the vaccine and have never had dengue infection actually lead to dengue infection. So they actually become sicker. And they recommend people getting dengue first before actually using the vaccine. Unfortunately, there is a vaccine available for the yellow fever virus. So, as I mentioned, these were considered just tropical diseases, third world diseases. But looking at the United States, the CDC comes up with these nice little maps, and they actually track the mosquito very well, and also dengue, Zika, and chikungunya infections. And so what we're looking at here is the estimated potential range of the Aedes aegypti mosquito in the United States. And yes, this is a 2017 map, but it takes a while for them to collect all the data from all 50 states to come up with such a predictive model. But what I want to point out is that even though it is a predictive model, they actually take county level records, historical records, and climate variables to actually come up with this map. So what we're looking at here then is the mosquito's ability to live and reproduce. So in the darker blue, you can see that we actually have a large area in which the mosquitoes can actually predicted to live and reproduce. And it's not surprising that the southern states, we actually see, they predict a large amount of the presence of the mosquito. And in fact, over the years, the CDC and the National Resources Defense Council, for example, have actually already reported the presence of the Aedes aegypti mosquito. And so then in the light blue, you can actually see then that the likely presence of the mosquito to live and reproduce. And then the lighter blue is the unlikely, and then the white is very unlikely. But the reason I wanted to bring this up is because when I actually started looking for positions back in 2013, I used this map right here. This map is a 2010 map from the National Resources Defense Council, or the NRDC, showing the presence of counties in maroon, as you can see here in maroon, showing counties that actually reported the presence of both the Aedes aegypti mosquito and the Aedes albopictus, another Aedes species that can actually carry the dengue and chikungunya, Zika, and yellow fever viruses. So what I wanted you to really, get from this figure is that in the maroon area, look how many counties were actually reporting the presence of the mosquitoes. And you can also see the number of cases of dengue in this case. But compare that now to the predictive model from 2017. So the maroon now, it's the darker blue. It actually covers all the southern states, all the way from Ohio, Indiana, Missouri, Kansas, up to California, where in 2010 that wasn't the issue. Where now, we only had certain cases, it's probably close to the Central Valley, but now we're actually extended even further. And in addition, this light blue gray area were areas predicted to be vulnerable to dengue infection. Well, that area also increased, as you can see in the lighter blue here on the map on the top. So I don't want to be an alarmist or anything. And also, for my meteorology colleagues and friends, if people don't believe you that, global warming is a myth or all that, tell them to follow the mosquito, because the mosquito, she prefers tropical weather. She prefers warm weather. And since the climate is getting warmer, the mosquitoes actually traveling further north, where now it's actually warmer and the mosquito can flourish no problem. So again, I don't mean to sound to be an alarmist or scare you or anything, but we are doing something about this because this is very relevant to us now. This is super relevant to us in the United States. It's no longer a third world problem. So what we're doing about this is I'm going to tell you about two projects that are ongoing in the lab, and these are actually pretty big, broad projects that I'm going to just focus on to kind of give you an idea of what we're trying to do. So to help us in our quests, one of the projects that we're focusing on is trying to develop mosquito selective inhibitors against blood meal metabolism. So if you remember from the first slide, I actually told you that these proteases or these digestive enzymes are expressed to help break down blood meal proteins. And then once she has those nutrients and that energy, she can lay eggs. And so the idea here is, and if we can stop this, we could potentially then stop the egg laying process. In the second project that I want to talk about today, focuses more on the eggs themselves. So the mosquito will have these beautiful ovaries, and she will actually lay these white little ovaries in an aquatic environment. And then through a process known as melanization, what's going to happen is that the outer shell will become nice and pigmented and dark, but it's also going to be forming a really tight, a hard outer shell that will prevent any water or liquid from escaping. So essentially, what you're seeing here in this figure is this beautiful little white egg laid in an aquatic environment, which allows water passage through to actually bring some water into the embryo. Then, after melanization, that embryo is protected because now there's nice moisture in here, and this egg can live anywhere from 6 months to even more if there's no water around. So what I'm going to be focused on is first starting off with the first project, the mosquito selective inhibitors against blood meal metabolism. So as I was telling you, there's a possibility that we could target midgut proteases. Proteases are actually very robust, and in fact, they're actually therapeutics that target proteases. And a good example is HIV-1 protease. There's actually good protease inhibitors that target HIV. So we were thinking, well, let's take this same idea. Can we target midgut proteases? So from work from graduate school, we decided to conduct this experiment where, let's see, can we limit the amount of nutrients or the amount of blood in a blood meal and then determine what effect that's going to have on the ovaries or the eggs laid. And so what you're seeing here is we actually conducted an experiment where we diluted blood meal in a feeding buffer from 80 to 20% blood meal content. And so that means that we limited the amount of blood meal proteins, which means that there's not going to be enough nutrients for the mosquito to actually make enough eggs. So one female Aedes aegypti mosquito, after being fully engorged, can lay anywhere from 100 to 200 eggs. And you can see in the 100% area that there's a lot of nice little ovaries. But as you dilute the blood meal, you can actually see that these nice little oval shaped eggs are getting smaller, smaller, and more deformed. And in fact, the mosquito will actually not waste this amount of protein. She can recycle this and then try to see if she can actually capture more. So then the idea here was, okay, we could potentially then target proteases to have a similar effect. But before we could do this, we need to identify what proteases are involved in blood meal digestion. So since the 1950s, it was known that digestion in the midgut mosquito actually is a biphasic process in which there's an early phase of proteolytic activity followed by a larger phase and more increase in proteolytic activity. But no one actually quantified this, and we wanted something that we could quantify so that way that when we conducted our experiments, we can actually measure that activity to see if we actually had an effect on protease activity. So to do this, what we did is we actually fed mosquitoes bovine blood, cow blood, and we dissected them at different time points over 48 hours, and we measured the total activity, the total proteolytic activity, and we came up with this nice quantifiable plot that you see here. And that way we can actually then have something to quantify. And whenever we had an experiment where we either inhibited the protease or did a knockdown experiment, we can actually determine the effect of that. But now going back, what proteases were identified? Well, in the 90s, the early phase proteases that were known were Aedes aegypti early trypsin, as you can see here, Aedes aegypti chymotrypsin, Aedes aegypti serine protease 6, serine protease 7, and late trypsin. So these were actually very well known in the 1990s. And then later in the 2000s, you have another chymotrypsin protease, JHA15, and serine protease 1 that were later identified. And this is not an exhaustive list yet. But before I go on, I kept talking about proteases and proteases and and digestive enzymes. But what is a protease? So hopefully a lot of my biochem students are watching this, and some of you who don't know what a protease is, hopefully you'll know what a protease is and then know what kind of activity this enzyme can actually conduct. So a protease is an enzyme that catalyzes cleavage of peptide bonds. So peptide bonds are actually formed when two amino acids are linked together. So an amino acid will have an amino group, as you can see in the nitrogen side, and then a carboxylate group with the CO group. And the nice thing is that you can actually link the carboxylate group with a nitrogen group, forming this peptide bond. So this will actually help link amino acids together, so that way you can actually get your nice proteins, and your enzymes and whatever else is made of proteins are linked through these peptide bonds. But also, in addition, this carbon here from this carbonyl group is susceptible to catalytic attack, so actually it could break up this peptide bond, releasing the amino acids. So there's two types of proteases. You have the endoproteases that actually cleave peptide bonds within a long polypeptide chain or even a globular protein. These endoproteases will break it down into smaller polypeptides. And then you also have the exoproteases that focus on the external or at the ends of um, polypeptide chains. So you have aminopeptidases that focus on the amino groups, the nitrogen side, and then carboxypeptidases that focus on the carboxylate side. But in the case with midgut mosquito proteases, we're dealing with endoproteases known as serine proteases. And so these serine proteases use the amino acid serine as pretty much the one that attacks that carbonyl that I mentioned in the previous slide. And this helps conduct the chemistry to break up those peptide bonds. And furthermore, you can classify many of these serine proteases into trypsin like proteases, where they prefer lysine and arginine, serine collagenase proteases, where links of glycine and proline form collagen and they're really packed together, but there are specific proteases that can cleave and degrade collagen. And then chymotrypsin like proteases, phenylalanine, tyrosine, and tryptophan are the preferred groups for these specific enzymes. So now that I've told you a little bit about the proteases, going back, early trypsin is one of these trypsin-like proteases. SP6, SP7 are also trypsin-like proteases. And then AA-chymo is a chymotrypsin-like protease as well with JHA15. And this one and late trypsin are believed to be serine collagenases, but that's a story for another time. But now that we have this information, we wanted to take a step further. Are these proteases directly involved in egg production? So we turn to a technique known as RNAi or RNA interference. So RNA interference is actually a conserved biological response to double-stranded RNA. So viruses are actually double-stranded RNA, and so that means that we're either going to have an infection or something's going to go on with these viruses in our bodies. So this conserved system is found in many eukaryotes, and luckily it's also found in the mosquito. So the way RNA interference works is that it takes advantage of the system in the cell where this huge complex known as a dicer complex will identify the double-stranded RNA, and then this will actually clip it to small interference RNA anywhere between 20 to 25 nucleotides long. And then another complex, the RNA inducing silencing complex, will come in, identify these little nucleotides. And the nice thing is that through activation with ATP, it will actually unwind the double stranded RNA. It will degrade one strand and then use the other strand as a template to identify other mRNA or other double-stranded RNA that's similar and then degrade it completely. So essentially what's going on here is you're silencing the gene. It's different from knocking out a gene where you completely eliminate the gene from the DNA. In this case, the gene is still present and it could eventually be transcribed and make mRNA, and then the mRNA can then eventually be made to protein. But with RNAi, what you're doing is you're eliminating that mRNA, essentially silencing the gene and not producing protein. So we did this and we designed the double-stranded RNA against early trypsin. SP6, SP7, and laid trypsin. We injected the mosquitoes, we blood fed them, and then we allowed them to lay eggs. So what we're seeing here then are the effects of this experiment where we allowed these mosquitoes to undergo a first gonotrophic cycle. So if you remember, that was the uptake of a blood meal, digestion of the blood meal proteins, and then the egg laying process. So what we're looking at here are individual mosquitoes and how many eggs those mosquitoes laid. So the F fluke, this is our control. So we wanted to make sure that the injection into the mosquito did not have an adverse effect that would actually mimic the effect of essentially knocking down the genes. So since the mosquito does not contain the firefly luciferase gene, we designed double-stranded RNA against that and then injected this into the mosquito. And this was important because the mosquito is a eukaryote. And so that means that it will have an immune response to the injection site. And so we wanted to make sure that injecting this did not have an overall effect that could skew our results. And luckily for us, the control matches almost nearly identical to our wild type um, mosquitoes. So we didn't include that in here, but we just wanted to have the control and show you that the injection does not affect any processes. But when we knocked down SP6, SP7, and laid trypsin, we did see a decrease. And this is actually a very significant decrease in the amount of eggs laid. Um, but notice that when we did our mix, we only saw a 31% decrease in the amount of eggs laid. So this was very interesting to us. And so we allowed the mosquitoes to go enter a second gonotrophic cycle. But in this case, we only took the mosquitoes that were fully engorged just to measure, if there was any effect of the double-stranded RNA left or if we needed to re-inject. And luckily, that double-stranded RNA effect was still present even in the second gonotrophic cycle, which led to a decrease in the amount of eggs laid. But this question was, why isn't there a synergistic effect? If SP6, SP7, and laid trypsin did have an individual effect that was significant, why when mixing them together, we didn't have a huge synergistic effect? Well, as it turns out, as I mentioned in the previous slides, that there are an abundance of other midgut proteases that had not been identified. And so that's where we pretty much come into now here at San Jose State, is where we've identified all these other proteases, and now we need to actually study them in vitro. We need to isolate them, clone them, and then see if we can actually get activity of these proteases. So then we can tie this back to the biological relevance of each of these individual enzymes. And so here I'm going to kind of switch gears a little bit to tell you about the work that my students have done and the great work that they've done here at San Jose State. So one of the things that I haven't told you is that proteases, whether they come from the mosquito or they're found in humans, they have to be expressed as their inactive precursors known as zymogens. So for example, trypsin is called a trypsinogen when it's in its inactive form. And the reason this is very important is because if you have an active protease being expressed, that protease will digest whatever protein is near and can actually have a deleterious effect on the cell itself. So we want to make sure that the protease is in an active form, and then through activation, through this peptide linkage. So there's a little sequence called the activation peptide or propeptide region that gets cleaved, helping induce a conformational change to the active form of the protease. And now this actually opens up the active sites, binding pockets, and is very active to then cleave the protein of interest. So when I was in graduate school, I decided, okay, I'm going to clone these genes, the early trypsin, SP6, SP7, and late trypsin genes, and clone them, and that way I can express them and see if I can get activity. So typically, what I like to, what I used to, when I tell this story, I used to say that, I was a little embarrassed that, I took this approach in graduate school, but now I'm very proud that my students identified something very important. So one of the things is that when I clone this gene, it has this little sequence, this little red sequence that you can see on the slide called the leader or the signal peptide sequence. So every protein enzyme made in a eukaryotic cell will have this little a leader sequence that tells the cell where to ship it to in the correct compartment of the cell. So in the case of the mosquito proteases, it's going to go into the midgut. Well, when I first started here in 2013, I actually used the Chem 131B capstone biochem course, our lab biochem course, kind of as a tool to have students get an experience of actually conducting real biochemical research if they weren't in research labs already. So I decided to use my research and incorporate it into the class. And so my students were able to look in the literature and find that when you're trying to express eukaryotic enzymes or proteins into a bacterial expression system, that leader sequence can actually affect solubility or proper folding of the enzyme. So from that, we decided, okay, let's remove the leader sequence and clone it into the bacterial expression vector system. So that way we can take the mosquito enzyme and use bacteria to produce a highly abundant amount of enzyme, but also do this in a short period of time. So with cloning into the PET28 vector, we introduce a his tag to our enzymes, so that way we can help with solubility, with purity in one single step. Furthermore, one of my research students at the end of the semester noticed that the company New England Biolabs, they actually made new bacterial strains that were not available when I was in graduate school. And these are very relevant because in wild type E. coli, the cytoplasm, which is the site of transcription and translation of recombinant proteins, it's actually very reducing, and this actually plays an important role in the bacterial system. So if there's any type of oxidation reactions or anything that needs oxygen, will actually not be allowed to take place because of this reducing cytoplasm. So my student found these cells indicating that these cells have a more oxidizing cytoplasm because they remove these specific enzymes that keeps the cytoplasm reducing. And why this is relevant for us is because the mosquito proteases, many of them have a minimum of 3 disulfide bridges or 3 disulfide bonds. So these bonds are actually very important to help the structure and stability of an enzyme. So as you can see here in this figure at top, you have these little yellow circles. These are individual cysteine groups. So these cysteine groups, when the protein folds, it has to fold properly in its perfect conformation so they can actually form a covalent oxidative reaction bond here. And so without this, then the protease will not be soluble, it's not going to be folded properly, and it's probably going to be inactive. So my students did a great job of identifying these, and we were actually able to produce soluble proteases, soluble mosquito proteases in a bacterial system. And in fact, my students who started in 2013, and I apologize to some of my colleagues that I know I stole some of your students. They joined my lab later, but we were able to actually finally, after a lot of kinks, publish their work in 2018. And these are actually figures from the publication. So what we're looking at here is the soluble expression of the different proteases, early trypsin, 6, 7, and late trypsin, where we introduced the plasmid that has the gene of interest into bacterial cells. We grew them up in specific nutritious media, and then we grew them up at different temperatures, and we collected samples at different time points, and we analyzed those samples by running them on a protein gel. So for a total sample, for example, we spun down the pellet. So we took the bacterial lysate, we spun it down to get a bacterial pellet. We removed the supernatant, then we added buffer, and then we lysed this using sonication, so vibrations to actually lyse the cells. And we took a sample, and that was our total. And for our soluble sample, the idea here is that we centrifuge the total sample, and if the protein is properly folded, it's going to stay in solution. But if it's not properly folded, that means that the protein is insoluble and will aggregate with itself, but also the membrane and end up in the pellet. And so you could see in all of these figures that we actually were able to solubly express all of our proteases. And so the next step was to actually purify the protein. And if you remember, I mentioned that we included 6 histidines to help with the purification process. And so that actually did help us purify in a single step. And you can see with this gel figure on the right that we were able to really clean off everything else, as you can see in the figure on the left. So we were able to purify and isolate our protease using these conditions here. And the great thing is that this was actually the inactive form. So we were able to control the activity of these proteases so that we didn't lose it while we were actually purifying these enzymes. And so just to activate, all we had to do is activate via buffer exchange. It worked with early trypsin, SP7, SP6. And then we figured out that late trypsin was activated by SP6. And so this pretty much kind of summarizes a lot of the work that we did that my students and I were publishing, or we got published, and we're still focusing on the other midgut proteases that are present. So this is kind of still a continuing project and focusing on the proteases where we're going to repeat these recombinant expression and purification systems. So now what I'm going to do is I'm just going to switch over and tell you a little bit about the melanization process, the whole the work that we're actually conducting to help figure this out. Because in the literature, if you actually look for information on eggshell melanization, there are a lot of papers out there focusing on proteins and enzymes that are involved in helping this beautiful little white egg go from this allowing water to come through and then provide moisture to the embryo to the melanized, hardened outer shell to protect the embryo. But the thing is that proteases are an afterthought in this. And so we want to go after this and we want to see what specific role proteases play in this process. And also, are they essential? Could it be that just one protease is involved in helping melanize, or are there a series of proteases that are involved at different steps? So that we're hoping then that with our work here at San Jose State and our collaborators at University of Arizona and their biological studies will help answer these questions. So to kind of set this up, what we did in collaboration with the University of Arizona, we wanted to determine what proteins were actually essential for the eggshell process. So we performed bioinformatic analysis, meaning that we actually analyzed a lot of proteins and enzymes that were predicted to be involved in eggshell melanization. But in addition, though, we wanted to be specific. We wanted to identify only those that were mosquito specific, that were not found in other organisms and other species at all, because we don't want to have any adverse effects if we do manage to make an inhibitor. And so the two proteins that I'm just going to briefly go over is EOF1 and CAD-L3. This one is called eggshell organizing factor 1 and cathepsin L3. So for these studies, once we identified the specific proteins, we used RNAi similar to what I discussed with the midgut proteases. We took fresh mosquitoes. After 1 day of eclosion, we injected them with double-stranded RNA. We let them go through the first gonotrophic cycle, the first oviposition, a second, and a third. I wanted to show you the effects of this experiment, which is something that we were totally not expecting. So when we compare the fully melanized eggs from our RNA F-loop control in the first, second, and third gonotrophic cycles, you can actually see nicely melanized eggs. But with our knockdown, a lot of these eggs are not melanized and they stay nice and white. And furthermore, in our second and third gonotrophic cycle, that double-stranded RNA is still maintained, still giving the same results. So this was very great for us. That it was like, okay, we found a specific protein that we could possibly target. And so for CAD-L3, we actually saw a similar effect. But notice that in this figure, it looks like there's maybe a mixture, about 50% of melanized and non-melanized eggs. And in fact, we did quantify, as you can see in this little graph here, we quantified how many eggs were melanized from this experiment. And compared to our F-loop control, there's 100% melanization. And then our CAD-L3, there's about 55%, which is still a good number, right, if you think about it globally. But we wanted to measure viability. It's like, are these, half of these melanized eggs, are they viable? And to our surprise, only about 5% of those eggs were viable. So this is great, meaning that this could potentially be a target to actually make a specific inhibitor. So that way we can decrease the amount of viable eggs in eggshell, meaning that this will be focusing on the eggshell and water only, and we don't actually have to go into the mosquito so much. But now is nothing is known about this protease. All we know is that it's a type of cysteine protease. So what we want to do now is we wanted to clone the gene. And so I actually took advantage of my Chem 131B class again in spring 2018. And they cloned the gene initially. And one of my students from the class, Mr. Aditya Malekar here, he decided that he wanted to continue the project and he decided to join our lab as a master's student. And so he was able to actually solubly express the protease. So what we're looking at here is a similar growth experiment of our cathepsin enzyme here. But what we did here initially is we stained the gel. These are the same gels, but we stained it with a specific stain called histag stain. So remember that our proteases have these 6 histidines. So we wanted to use this also as a marker because if you look at the gel here on the right, we really can't tell that where this arrow is pointing to is actually our protein of interest. But with the histane, we can actually see that our protease actually was produced nicely. So Aditya was able to solubly express this, and he did some initial purification experiments and things like that. And we sent some samples off to UC San Diego to see if we can determine what kind of activity this protease might potentially have. And so to summarize, what we really want to focus on is really biochemically characterizing these proteases. We need to go through these in vitro studies, isolate the enzymes, determine what kind of activity they have, determine the structures, so that way we can actually focus on making mosquito selective inhibitors, either to go and focus on the blood meal digestion proteases or eggshell melanization proteases. Because if we could do this, then we could potentially create a new vector control strategy where hopefully in this quest will lead us to minimizing the mosquito population and then also minimize pathogen transmission. Because if there are potentially hardly any mosquitoes, that should potentially minimize pathogen transmission. So this is the end of my talk. And before I finish, I just wanted to give, some quick acknowledgments to my students. They're the reason why I have been very successful here at San Jose State. We've been able to get funding from the NIH, and this April, I'm happy to report that we actually got for 4 more years of funding. My renewal got approved, and we were awarded in April, and it was because of their great work. The students here that are in teal, they're the ones who were authors in the publication, and I'm hoping that we can continue this on. So I wanted to thank them, and I also want to thank you all for being here, and thank everyone else too for inviting me and giving me an opportunity to share my work with everyone. So thank you very much.
Awesome. Well, thank you so much, Alberto. We're already starting to generate some questions. We've got some time for them. I have just a quick one. I'm going to take chair's prerogative, I guess, and ask as the first one. One of the kind of assumptions is, where mosquitoes live, and those maps early on, show high desert areas and so forth. So, question is kind of they need water. What is the relationship with kind of human life and the expansion of this mosquito population? You talk about climate change, but what role does like standing water and things like that play in the expansion of these vectors?
Yeah, and that's actually a very great question because one of the things that I didn't mention is that the Aedes aegypti mosquito is an urban vector. And specifically, she prefers to live with humans. Why? And I hate to say it like this, is because we're dirty. We drop trash everywhere. So because of that, there's more little places where water can be accumulated or collected. And so mosquitoes will take advantage of that. And so that's why if you looked at the map, as you mentioned, right, desert areas and things like that, but if there's people there, there's going to be trash and then water can collect pretty easily. And so that's why a lot of mosquito control agencies always tell you if you have a bird feeder or anything like that, clean it out. Don't give mosquitoes too much access to the water, change out the water or dump it out or something, especially during the warm weathers, because the mosquito will take advantage of any little water present. And that's the I think I'm in love with this mosquito, right? Because it is a killer, but at the same time, you know, the evolution of taking advantage of just little water that's present anywhere, it's what amazes me of this little creature. These eggs are so robust that they can survive. So if they were, like when we were in Arizona, the mosquitoes, yeah, we're gonna have dry seasons, and that little egg can live no problem until there's a water source, and then boom, we'll have a flush of mosquitoes, and then we have a whole life cycle, and then we could keep going.
Yeah.
So yeah, it's a great question.
Yeah, thank you. So another question that came in: what role do mosquitoes play in the terrestrial food web, and what are the potential impacts if they were eradicated?
So see, that's another part of this discussion that's very important, because as I mentioned, the Aedes aegypti mosquito is an urban mosquito. She prefers to live with us. So there's already thousands of mosquito species out there, thousands, but this, only a handful of mosquito species live with us. And in reality, that really doesn't affect the whole ecosystem of the other thousands of mosquito species that are out there. So the thing is with us, it actually will benefit more for us because they prefer to live with humans. And there's usually, I mean, there's some predators, but we try to kill all other bugs anyway, right? So, but in this case, since there are thousands of mosquito species out there, that only these handful will not have a huge effect on the ecological system.
Um, Another question that came in is why are there so many cases of dengue reported in northern states like Washington and Wisconsin? Which is outside that map, right, that you produced early on.
Right. So there, the first map that I showed didn't have a lot of the cases, but what I didn't mention was that a lot of these cases are cases that people have gone and reported from taking a vacation, going to South America, or these other countries that are endemic with the mosquito and the viruses. But the thing is that I don't know if many of you have kept up with the news, but Florida has always had a dengue problem, and the Key West has been brutalized with a lot of dengue, a lot of Zika. And just recently, the EPA approved the release of transgenic mosquitoes to try to help with that aspect of minimizing dengue infection. So a lot of these were just reported cases when people went on vacation. But as the map and the mosquito population is increasing, that means that there's also a possibility that it only takes one person to be infected and infect a mosquito, and then that will then lead almost like a domino effect.
So we often talk about the mosquito as a vector, but actually it's the human that's the problem here.
Yeah.
Okay, so I won't go down that trail for you. Those next question is the EOF1 protein specific to mosquitoes, or could there be some risk to the eggs of other species if this was used in the wild?
No, and this is the beauty of our experiment. And I think that's why PLOS Biology, when we submitted it, they almost immediately accepted it, is that we focused only on those enzymes and proteins that are very specific to the mosquito. So that's why I mean, it was an extensive bioinformatic analysis comparing other egg layers, especially the important pollinators like butterflies and bees, to make sure that we did not have any type of similar proteins. And this is actually very specific and unique to only the Aedes aegypti mosquito.
Well, that's a really interesting, and it segues nice, I think, into the next question, which is, what are the advantages of using the methods described here over pesticides and other strategies that have traditionally been used to mitigate or eliminate mosquito populations?
And yeah, definitely you're correct on being a great segue, because a lot of these insecticides do affect the good pollinators, butterflies, bees, other insects that can actually have a huge effect and impact on, the ecosystem. And so for us, this is why we've been trying to push more of being a more specific mosquito selective inhibitor, so it doesn't affect anything else. And that's one of the difficulties of trying to do this work. But now with advances in technology that we're able to do recombinant expression, we could clone something so easily. We can determine structures very easily now, as opposed to a few years ago, where that way we only specifically focus on the mosquito enzymes and nothing else. And of course, we want to go through trials to make sure that it doesn't affect other insects as well. And that's why we did an extensive study to make sure that that doesn't happen.
So another question that comes up that I think is really interesting in your talk is um, that HIV was a health, for lack of a better word, a helpful model for starting to ask the kind of questions that you ask. And I know HIV is a retrovirus, right? Early on, it changed the game, right? In the study, because retroviruses were fairly newly discovered in the 80s. And anyway, can you talk a little bit more about how HIV as a virus and so forth kind of impacted this work you're talking about here, or informed it, I guess is the right
Yeah,
way to put
definitely. So the nice thing, with the studies with the HIV virus is that um, at the beginning, they really didn't know how replication worked. How does this virus take advantage of our system? And it turns out that they actually have specific proteases that will help with the replication once it invades a human cell. So once the scientists determined that this was the case, they're like, you know what? It's a specific protease from the virus. Let's do exactly what we're doing. Let's isolate it. Let's study the protease. Let's do the structural studies. And sure enough, they were able to target specifically the protease in HIV. And so that's kind of a similar approach that we want to take, is especially with these viruses. Zika, dengue, chikungunya, they all take advantage of the mosquito cell, but also the human cell. And the nice thing is that they use specific proteases. They have specific proteases that will help with the processing and also with the infection once it's in the cell. So potentially with us, there's a possibility that maybe we can get to a point where we can target the specific proteases, right? And this is something that I would like to do in the future, is to get into the virus realm and see what else we could learn from the proteases there.
No, that's fantastic. I have to say, if you had been my first year chemistry professor, I might not have become an international relations and East Asian studies major in college. We did organic the first year, and it was all over for me after that. But I have to say that you're incredibly engaging. We got one more quick question before we send you off, and thank you. So you mentioned RNA knockout, but instead used RNA repression. Why not use RNA knockout?
So it's very difficult to actually knock out these. Well, let me rephrase that. It used to be difficult to knock out genes in a mosquito, but now with the CRISPR system, this is actually now something that we can do. And in fact, my collaborator at the University of Arizona, he's been dabbing with a little bit of the CRISPR to see if we can actually knock out some of these genes and go in that direction as well. But initially, it's very difficult to kind of manipulate the DNA in a living system. So that was kind of one of the drawbacks of trying to knock it out in the mosquito. But now, if we were using something simple like bacteria or something like that, yeah, that would be easy. But it's a complex eukaryote, so that made it a little difficult to do. But with CRISPR now, this is something that it actually opens the door for more studies and more specific studies too.
Well, I just have to say, Alberta, I really appreciate the opportunity to hear from you today. Congratulations on the extension of the NIH research, and congratulations to you and all your students. I mean, what's so amazing here is you show that importance, that collaboration work across institutions, right? That we need to have to answer these really big, wicked problems, but that students can play a really valuable role in all this. And you've had an amazing amount of success here at San Jose, and we really are thankful to have you here. So thank you so much for this fantastic talk and for engaging us today. It really was absolutely special. So thank you all as well for those who were able to make it here. And as a reminder, we'll have this out as a video recording so people like Carl Chemnitz can watch it. So thanks, Alberto.
You're welcome, and thank everyone too. Thank you all.